Well, the first thing we have to understand is this: the entire belief that genes turn on and off by themselves is a totally false statement. Because by acknowledging that, then you're saying, well, I had no control over it. The genes did it, okay? But we know, no, genes are blueprints. They're there to design what are called the proteins of the body, 100,000 different proteins. The proteins are like Lego pieces that you can assemble them, the 100,000 different proteins in different combinations, make a muscle cell with one combination of proteins, a nerve cell with a different combination already. So I say, yeah, but these are the proteins that create us, and these are the proteins where life comes from. I said, but what about the DNA? I said, well, proteins wear out. That they're called labile, not stabile. Labile meaning that they fall apart. You put a piece of meat on a plate and we come back next week, that meat is not going to be the same as it was when we put it out there. It's going to start to break down, fall apart. So proteins that naturally keep falling apart, but then you have to replace them. I say, yeah, but proteins are complex molecules with a complex structure. And I go, oh, I said, well, then how do I replace these complex structures? I say, there are blueprints. A blueprint to make this protein or that protein, 100,000 proteins. I say they're blueprints. I say, what do you call a blueprint? I go, a gene. A gene is a blueprint. I go, oh, a gene is a blueprint. And then we told people, well, they were self-emergent, which means, oh, they turn themselves on, they turn themselves off. I say, no, look, let's be honest about a blueprint. We go into an architect's office. She's working on a blueprint. You lean over her shoulder and you ask her, hey, is your blueprint on or off? And she'd look at you like, what the hell are you talking about? There's a blueprint. There's no on and off. I go, precisely. The difference is you can read a blueprint or not read a blueprint, but the blueprint doesn't read itself. And all of a sudden I said, then for years we were saying the blueprint was turning on and off by itself, controlling you. And now we know, no, it's the consciousness that engages the blueprint. I say, how does it do it? And I say, through chemistry. Different chemicals cause different responses in genes. Matter of fact, that's what I did in 1967, which is now like 40 or 50. It's a long time ago. And what did I do? I had genetically identical cells, cloning cells, and I had 3 plates with genetically identical cells in them, but I fed them different culture medium. I said, what's culture medium? Well, that's the growth environment. I go, yeah, but what is culture medium? I say, laboratory version of blood. So if I'm going to grow human cells, I say, what is human blood made out of? And then I go into the lab and with a recipe, I make culture medium match human blood. Okay. And so I go, so what? And I say, well, because I'm creating the mixture, I can change some of the composition. So I created 3 different combinations of chemistry called culture medium, slightly different. And I had 3 plates, genetically identical cells on all the plates. And what I did is I fed 1 plate with culture medium version 1, another plate with culture medium version 2, and a third with culture medium 3. And I said, well, what was the result? They were all genetically identical. I said, well, in one environment, culture 1, the cells form muscle. In environment 2, the cells form bone. In environment 3, the cells form fat cells. And I go, well, wait a minute. I'm teaching that genes control life. And here we have an experiment that reveals all these cells are genetically identical. Then what was the cause of why one becomes bone and one becomes muscle? And the answer was, it wasn't the cells, it was the environment, the culture medium. And I go, wow, that's really neat. That's the foundation of what we call epigenetics. The genes didn't control this. They were controlled by the environment that the cells were in, okay? So I go, so what's the significance for this? I go, well, a human, when we see ourselves in the mirror, single entity, and I go, well, that's an illusion because we're made out of 50 trillion cells. The cells are the living entity. Bruce, by definition, is a community of 50 trillion sentient cells. So I go, oh, so the cells in our body is like a skin-covered petri dish. It's got 50 trillion cells inside. I go, yeah, but it also has the original culture medium. I said, what was that? Blood. And I go, then what? And I say, it doesn't make a difference if a cell's in a plastic dish or the skin-covered dish. It's still controlled by the environment culture medium. In the plastic dish, that's the one I put in there. In your skin-covered dish, your blood is your culture medium. So bottom line is this. It's the chemistry of the blood, the environment, that activates expression of the gene. And I go, now comes the most, well, where's that chemistry come from? I go, the brain is the chemist.